chapter is called Medical Microbiology, which is kind of a vague chapter because it talks about a whole bunch of different things, which is why, again, it will be helpful if so what I did was I saved myself the table of contents that's a little bit more detailed, and I will post this in a reply to the week 2 lecture and 205 announcement. I might not get to it till tomorrow because I think they're as separate files and I got to figure out how to combine them into one file. But I will get that done in a PDF. Yeah, because I did type in the title names, but the title names are kind of vague for what they covers for some of them. So, okay, so medical microbiology is the study of the collection of samples from individuals. If somebody has an infection and they go into the doctor to try to figure out what type of infection they have, usually they're going to have to take a sample and they're probably going to have to send it out to a lab because a lot of times they don't actually have the equipment and the testing chemicals that they need right there in the office, which would be really helpful because it'd be a lot easier to treat the infection correctly. But so this chapter is kind of all associated with information about that. And then there's kind of a continuing one, the next chapter. So this chapter is going to focus on bacterial infections. And then the next chapter is going to focus on virology, mycology, and parasitology, which is basically just these different types of organisms or infectious agents. I have a couple of links in the PowerPoint that I posted up there, and there might be something that I added in this one. I can't remember which one it was, but I have a few links in there that are going to be helpful information. I'm trying to figure out, somebody's not on mute that had noise, so I'm trying to figure out his, oh, it's me. Get off of there. Okay. I'm talking to my computer. Okay, so healthcare associated infections is one of the main topics that's actually listed on your study guide. And I've added a couple of resources in here to give you some more information about that. But healthcare associated infections are also called nosocomial infections, which I don't know that that term is in your textbook, but that's another name for it that you're going to need to be familiar with. And these are any type of infection that is acquired while a patient is hospitalized or in a medical office. Infection acquired during hospitalization or treatment. So also could be like in a doctor's office or something like that. So that could be during a procedure, it could be during catheterization, it could be during wound treatment, it could be during a surgery. So it can be any activity that occurs while a patient is being cared for. And it's going to be because something was contaminated, something was not sterile that was supposed to be sterile. And then the infection is transferred via the clinician. So this link that I have here is to the Centers for Disease Control, and I basically just took a screenshot of, according to the Centers for Disease Control, the CDC, the most common sources for nosocomial infections. And this link here takes you to that website, and it has a little bit more information about each one. So this URL up here is what this picture is actually linked to. But these are some of the most common ones. There can be a sepsis that occurs through an infection in the bloodstream. There can be a urinary tract infection that is via a catheter. Those, the catheters and things like that, that are tubes, can have that slime layer that certain bacteria form, especially E. coli, which is the most common one for urinary tract infections that are nosocomial. It forms that slime layer that's kind of this goo that the bacteria form, certain strains of them form when they replicate, and it makes it really challenging for the antimicrobial agents to remove them. Of course, there can be surgical site infection. If you have anything that gets contaminated in your sterile field, you can have an infection there. And then through the ventilator, again, tubing, that is where you can get microbes that can get in there. These are the basic strategies in this kind of Venn diagram here for preventing nosocomial infections. But I also have a link to this website that is the best practices to prevent healthcare associated infections. So that can give you some more information on there if you want to read more about it. But basically, it kind of starts with hand hygiene. If you noticed on the previous one, it said hand washing properly is one of the best ways to prevent nosocomial infections. And then environmental hygiene, just making sure that everything is cleaned and disinfected sufficiently. And then also, not setting something down on a non-sterile area if it is sterile. Screening and cohorting patients, that is, checking to make sure individuals don't have other infections in certain areas of treatment areas where they could be spreading those to other patients. And then tracking that hospital-wide at some level, which is what the surveillance part says. Antibiotic stewardship, we're going to talk a little bit more about this later, but the problem of antibiotic resistance is a huge problem. And actually, I mentioned I was going to post one of those. I'll post it to the announcements. There's a quick facts sheet that I have on how clinicians can prevent antibiotic resistance and how patients can prevent antibiotic resistance, which is when the bacteria mutate to be able to not be killed by an antibiotic that would normally kill it. And that occurs, I can't remember whether we had this discussion in this class or it was my other one, so I'm going to mention it again. So I'm sorry if I'm repeating it, but can be by not finishing a regimen of a bacterial or antibiotic prescription. It can be the doctor prescribing an antibiotic when the patient actually has a viral infection. Antibiotics do absolutely nothing for a viral infection. So those are two of the major ones, is making sure somebody's taking the correct antibiotic, making sure that they're finishing the regimen, and then not taking an antibiotic when it's not beneficial. It's not going to help. Following guidelines, and then safety culture, which is kind of self-explanatory on those. So like I said, this link, if you want to read more about that, because I don't know how much detail it goes into in your textbook, it kind of gives you more information about specific steps that you can take to prevent nosocomial infections. All right, there's a small little section on the history of microbiology, and there's some key researchers or scientists that are usually highlighted as some of the most important discoveries in microbiology. And so what I did was kind of add in focus on the ones that are usually highlighted as the most important. Von Leeuwenhoek is considered to be the first to develop the microscope, although I've seen different scientists as that in every single textbook I think I've looked at. So it's kind of arguable, but he's definitely one that's known for one of the first microscopes that were being developed. And then I have a chart down here. I'm actually going to skip this one because it's kind of just silly definitions. That includes some of the other major discoveries. And we're going to do a little activity with this tomorrow as well in the Kahoot review that we're going to do. But Louis Pasteur, there was a few things actually that he discovered. He discovered that bacteria were responsible for fermenting things like beer and wine and stuff like that. He also disproved spontaneous generation, and then he's the one that came up with pasteurization, which is heating something to decrease the amount of microbes that is present in it. I believe it's in week 4, you're actually going to have a lab on sterilization and pasteurization. And it's really a good lab. We actually didn't have that one in our curriculum last session, and I'm glad they added it because it's really, really helpful for giving you a better understanding of the antimicrobial agents. Lister was responsible for figuring out that you could use phenol to sterilize your equipment before surgery, and that is what gave aseptic surgery, and they found that the number of patients that were getting infections after surgery drastically decreased. And that's where the name Listerine comes from, if that helps you to remember that. Robert Koch is the one that developed the germ theory of disease, and he's the one that came up with Koch's postulates, which are a specific set of steps that are necessary in order to prove that a specific pathogen causes a specific disease. Nicer, and you'll see this here a lot of times, the person's name sometimes might be in the actual name of the organism. So almost said developed gonorrhea. Discovered the pathogen, the bacteria that causes gonorrhea. And then I don't really hear too much about Finlay, but those are some of the major ones that you need to be familiar with. I did post actually in, move this out of the way real quick, in the week 2 lecture in 205 announcement that I just posted today. I posted the table from your textbook because it's really actually a big table and it hardly shows any of it when you're reading through and clicking in the ebook. And so I actually printed the whole thing to a PDF because it makes it a little bit easier to see. But this one goes into a lot more detail as far as the different individuals that discovered different things. Okay, and then part of this idea of what Louis Pasteur discovered is that he disproved spontaneous generation. This is one of the key things that you need to know from this material for probably several different chapters, is that spontaneous generation was the idea that organisms or cells just crystallized out of nothing and they would just spontaneously arrive. And part of Louis Pasteur's work that he discovered that says disproves spontaneous generation is that all cells come from pre-existing cells. You may have learned that in your Gen Bio class because that was one of the 3 tenets of cell theory, is that the cell is the smallest organizational unit of an organism, that all living things consist of at least one cell, and then the third part is that all cells come from pre-existing cells. And the mechanism for replication in bacteria, as you can see here as prokaryotes, so includes prokaryotes, which is bacteria and archaea, replicate by binary fission, which is much less complicated than the mitosis process that cell replication occurs in eukaryotes. And it basically has to do with the fact that there is no nuclear membrane surrounding the genetic material, and the chromosome is just one circular piece of chromosome. So this crazy purple blob here is really actually just one circular chromosome. So for binary fission in bacteria, that one circular chromosome is just copied, and then the cell just basically pinches into 2, which is what fission means. Fission means to pinch apart or to break apart. And then binary is the part that's saying there's 2 products. So a single cell copies the DNA, and then it basically just pinches into, and that's binary fission. All right, so for the section on naming bacteria, you need to know binomial nomenclature, which you also may have learned in your Gen Bio class, but we'll just kind of go over it again just in case. So for example, that Neisseria gonorrhoeae, that is the name of the bacteria. That is, the genus is the Neisseria, the gonorrhoeae is the species. So the official way that you do binomial nomenclature is the genus is first, and then the species is second. The genus is capitalized, the species is lowercase, and then they are both in italics. So just to kind of go back and show you that one. So you can see here, the Neisseria gonorrhoeae is the genus, Neisseria gonorrhoeae is the species. Some textbooks also refer to the species as an epithet. So if you ever see that term, that means the same thing as species. So when we go through and we learn about the different bacteria that cause the different diseases as we go through the different chapters, you're going to see this binomial nomenclature, which is the genus first and the species second. Okay, let's see here. This section, we kind of talked about this last time. We talked about the bacterial structure, and we kind of got a preview of this. We mentioned that they do not have a nucleus surrounding the genetic material. They do not have, which is instead called the nucleoid region. They have, some of them have a glycocalyx, which is the capsule, which you can see here. Some form what we call a slime layer, some a capsule. They're both made of a carbohydrate substance, which is why they're called the glycocalyx. So that is just kind of this extra sticky, you can kind of think of it like a sugar. If you think of a carbohydrate, a sugar is sticky. It's kind of this sticky carbohydrate layer that they secrete that protects them from the macrophages and other harsh environments. Not all bacteria have or form a capsule or slime layer, just some do. Some have a flagella like we talked about before, and some can form spores, which are those dormant ones that we talked about. And we'll talk about the specific ones as we go through the different chapters. So this is the picture that we looked at last time, and we kind of walked through all of the different structures. And then there's three basic shapes for the actual shape of the cell of the bacteria, of a single cell. And these are terms that you're going to need to know. Cocci is their round or sphere shaped. Bacilli are rod shaped. They kind of look like a little hot dog, depending on what type of rod they form. And then the sporilla are spiral shaped. And there are some variations on these, and you'll see more than just these three names for the shapes of the bacteria, but these are the three basic shapes. In your lab last week, actually, you also had Vibrio, which is like a comma shaped. It's basically a hybrid between a bacilli and a spiral. So it's kind of a bent spiral shape, broad, which is why I pulled that section up in your lab info that we went over. All right, so on this diagram here, I added in, this has the Vibrio that you saw in the lab last week. So a bacillus here is just this rod that's shaped like that. And then you're going to have your sporillum, your Vibrio, and then also a spirochete, which is basically a spiral-shaped bacteria that has a tighter corkscrew. Oftentimes, the genus or species of the name of a bacteria will have the shape of the bacteria in it. And then in addition to the shape of a single bacteria, so say for example, this Diplococci here, this is one bacteria, and after it replicates by binary fission, there's now two bacteria here. And they tend to stick together in those twos when they replicate. This one here, Strepto, means that it forms chains. So that is what that term is. So this is a chain of cocci bacteria. So for this particular, like that forms strep throat, for example, and you can see a micrograph of it in here. Each one of these is a single bacteria, that this whole chain of them stick together when they replicate. These terms, strepto for chain, diplo, for example, sarcinae is when like they form this cube-like structure of 8 that tend to stick together. And then staphylo, which you've probably heard before because of Staphylococcus aureus. Staphylo means that they form these grape-like clusters. These terms, staphylo, sarcinae, tetrad, strepto, are referred to as the cell arrangement. So it's different than, that is 2 R's. It's different than the shape of a single bacteria. It's how they congregate together when they replicate. That's referred to as the cell arrangement. Both of these things you can only see under a microscope in order to try to identify them. All right, I think I'm going to stop here and we're going to take a 10-minute break, and then we will come back and we're going to get into the staining, and then we're going to do a Kahoot to start learning that information, or just test your understanding after we cover it. So let me pause the recording. Okay, so we were talking, oh, whoops. Oh, Marla, can I make the PowerPoint full screen? I think if I open it in PowerPoint, I can, instead of the PDF. The PDF doesn't shape it correctly. That's fine. I already closed it, so it's not that big of a deal. Okay, so we're going to continue talking about the different shapes and the arrangements of the bacteria, and then we're going to get into the different staining techniques. And we're actually going to spend most of our time on the gram staining technique, because that is the most widely used staining technique for staining bacteria in labs. Basically, the first thing that somebody would do if they went in, had an infection, they took a sample from the lab, they would do that. What's going on here? I gotta move the chat box for a second for me to do this. That's the problem with having the PowerPoint full screen, is that I have too many boxes hiding everything on my screen, so it makes it hard for me to see where stuff is. Why is this? Okay, that was weird. That is not the one we were on. Is that better? Yes, okay, good. Okay, so the cocci, like we were talking about, this kind of gives you a better explanation. Like I mentioned that I couldn't find my annotate, so the cell arrangement again is how the single bacteria stick together basically when they're replicating, whereas the shape of the bacteria is the cocci, bacilli, or spirillum or spirochete. So just like we saw in the previous one, I think the ones that are the hardest for students to remember are the strepto, which forms chains where they're kind of end to end like a chain link fence, and then the staphylo, which are the grape-like clusters. And I think it's just because they're kind of similar words. So you just have to memorize strepto's chains, staphylo is clusters for that one. Now I put my boxes in the opposite place, and now I'm getting keep clicking in the wrong place. Okay, whoops, gosh darn it. Okay, how do I go back? Oh my gosh. I'm not used to it being a slideshow now. I forgot where all the buttons are. Okay, your rod-shaped again are going to be your bacilli. Many of the rod-shaped ones do possess flagella. And again, these can be in pairs, which would be diplobacillus, or it can be in chains, which would be streptobacillus. So you can have streptococci, that is a chain of sphere-shaped ones, or you can have bacillus, which is a chain, kind of like little sausage links on a chain of rod-shaped bacteria. And there are many bacilli bacteria that can form spores. So those are the most common shape that form spores. Whoops, I almost ended the meeting. See, I shouldn't have put the box. I keep clicking on it. I usually have my meeting controls on the bottom and my annotate on the top, and I switched them, and now I can't figure out which is where. That's okay. All right, spirula is, so spirula is plural, spirillum would be singular. The spirochetes are the ones that are this tightly wound, spiral-shaped bacteria. An example of this one is that cause syphilis is one of the examples, and Lyme disease. So we'll talk more about those specific ones later. Okay, there's one little section in here on guidelines for specimen collection, and a lot of this centers around making sure that you are using aseptic technique when you are collecting the sample, when you're transferring the sample, when you're doing basically touching everything. So key points for this are to collect the sample before the administration of antibiotics. You want to take it at the very beginning so that it has the actual bacteria that are there in the infection before any of them start to be killed. You want to collect without contaminating the specimen. The whole point of collecting a specimen is to try to figure out which one is there. And if you contaminate it when you're collecting the specimen, you're not going to know whether the contamination was what the infection is or whether it was something that got transferred to the sample. So obviously that's very important. You want to obtain sufficient quantity of the specimen from the site and make sure that it does not invade too much into the surrounding tissues, because you want to try to get as pure and as much of the culture of that one specimen. And then obviously place it in the appropriate container or transport medium so that it is maintained and makes it to the lab to be tested. And then, of course, label each specimen correctly. You don't want to get specimens mixed up. That would be silly. And transport it within the allotted time. So this will allow for the more accurate results to make sure that you can try to align the correct medication with the infection that's going to be most effective in treating it and most specific in treating it. And then prevention of the spread to other patients or other care workers. Oh, Marlo said your GYN office put the sperm sample in the wrong container, so they were all dead by the time they got to the lab. Oops. Very important. I'm sure some of you have seen on the news, they're saying that some of the vaccine hasn't been administered in quick enough time and they're having to throw some of it out. So yeah. So yeah, very important. Okay, this last section we're going to talk about tonight is bacterial staining. And I think I mentioned this before, but when you look at a bacteria or any cell type underneath a microscope, they're actually transparent. They're colorless. And so it makes it really hard to actually see any structures if it's just clear. It looks like a bubble under the microscope, quite honestly. So the process of staining allows us to be able to visualize structures and cells that we wouldn't normally be able to see, and then can also provide information about the classification. I mentioned this before, and I think it's in the lab 2 tomorrow. We're going to look at some actual pictures of cells under the microscope at 1,000 times magnification from my previous lab when we last had live lab, and they're so small that you really can't see any information. Not only that, this is a startling statistic. Okay, so there are estimated to be 1 million species of bacteria. And we have only characterized and named about 6,000. This was a few years ago when I saw these stats, so it might be more than that now, but 6,000 named and characterized to date. Even with 6,000, when you have 3 basic shapes of bacteria, and there's 6,000 different species that fit into those 3 basic shapes, you cannot tell by just looking under a microscope, even if you've stained it enough to see the shape of the bacteria, what species it is. And so there are all kinds of other chemical tests. We grow it on different types of growth medium that have different identifiers in them that are indirect ways that we figure out what species the bacteria is. But the gram staining is the first step, which is going to say they're either in this group or in this group. So it helps to narrow it down. And then we can do further tests in order to figure it out. So there's some stain names that are named on how or what type of information they give. A simple stain is just like it sounds. It's one color, it stains some structure, and it allows you to be able to see the structure. So it's just one step. You put the stain on and then you can actually see it. Then there are differential stains, which is, for example, like what the gram stain is, that just like it sounds, will differentiate 2 different types of bacteria. So we'll learn today the gram stain in gram-positive bacteria are going to stain purple, gram-negative bacteria are going to stain pink. And so these differential stains can allow us to distinguish. So say that we have a culture, a sample from a patient, and we're trying to distinguish which species are present. If we do the gram stain, that will identify whether there's a gram-positive and a gram-negative bacteria that are in that sample that have infected the patient. And then this one doesn't have it in here labeled like this, but in the previous textbook, there was a third type that's called a special stain. Oops. So I'm going to throw it in there just in case you need to know that one. A special stain is to stain cells that have structures that don't stain by these normal methods, by the simple staining or the differential method. And these are going to be your capsule stain. The flagella have to be stained with a special stain. There's acid fast, which are these certain types of bacteria that have this waxy covering of mycolic acid on the outside, and they don't stain with a simple stain or a differential stain. So those are 3 others. I actually should have put them in the PowerPoint, but I didn't think to add it. So you might want to add that in there. Special stains, examples of special stains are capsule staining, flagella, and acid fast. There's just some structure on the bacteria that causes them to not stain by the traditional methods that we use. Okay, one of the things that we do when we are staining bacteria, and one of the downsides of staining a sample is that it has to be killed in order for it to be stained. And one of the first things that we're going to do when we put the specimen on the slide is we call it fixing it to the slide. And we do this by heating it and running the slide through the Bunsen burner relatively quickly. You don't want it to incinerate the bacteria, but you want it to dry them out, and it's going to kill them. The reason we have to fix them to the slide is because you don't want the bacteria to wash off when you put the stain on there. there is quite a bit of washing in the gram staining process, and you're going to need to know what stain is for each step and what the purpose is for each of the steps. For your lab tomorrow, you're going to actually have to know how long each step is applied to it, and you're going to have to know that it's washed with water between each step. So the first step is to stain with crystal violet, which is the purple stain. Then, there's different protocols for how long you leave the stain on in different labs. I think I've been in 3 different labs where we've done it, and there are different times for each of them. So when you're in your labster tomorrow, and I'll mention this again tomorrow, you're going to need to read the theory tab to see the timing that it recommends in the labster, because you're going to have to put the correct times into the slots. But you wash with water after each step. So crystal violet's what we call the primary stain. And then you flood it with grams iodine, which is called a mordant, which you can kind of think of it like a glue that holds the primary stain on the gram-positive bacteria. So glue, it's not really glue, but it's like a glue, holds the crystal violet. I'm going to abbreviate that CV so that it fits on here. Crystal violet on the gram-positive. So grams iodine is a mordant. There are all different types of mordants, but that's the function that grams iodine serves in the gram staining process. And it actually doesn't even say it on here, but then you wash again with water after the grams iodine as well. I didn't notice that step was missing there. Then you're going to apply the decolorizer. You can either for this step use alcohol or acetone. Just depends on the protocol for the lab. It is the decolorizing rinser, which is going to wash the crystal violet off the gram-negative bacteria. So they will then be colorless. And we're going to look at a diagram here that kind of walks through these steps. And then the fourth step, there's 4 steps: the crystal violet, the grams iodine, the decolorizer, and then the counter stain is the second or last step, 4th step. And I don't know why it says red here, because it's actually pink that it appears on the bacteria. So I don't want you to get confused with that. But the counter stain, or secondary stain as it's also referred to, is safranin, which is a pink stain. It appears pink. So this picture here is showing you these 2 types of bacteria here. So the gram-positive bacteria in this picture are cocci, which is the little spear-shaped ones. And you can kind of see they look like streptococci because they're in these little chains. And then the gram-negative bacteria are these rod-shaped pink ones. So there's several things you need to know about the gram stain. You need to know which ones are pink and which ones are purple. You need to know what the steps are and the staining method. You need to know also what color each group of bacteria would be in each step, which is shown in this diagram that I have here on the next slide. So the answer key here is showing you what's in each step. So you have your crystal violet first, that's purple, then you have your grams iodine, then you have your decolorizer, then you have your counter stain. And then in the Petri dish on these pictures, it is showing you, so I want this circle. So, or it's not a Petri dish. It's basically just a field of view under the microscope. If you were to look under the microscope, you would see the cells. And for demonstration purposes, this one is showing you the gram-negative are these bacilli, and the gram-positive are cocci. But they can be the other way around. Not all cocci bacteria are gram-positive, and not all bacilli are gram-negative. So it's just showing you a different cell shape just for demonstration purposes to be able to show which is which. So in the first step, all of the cells are going to appear purple, both gram-positive and gram-negative. In the second step, they're still both going to be purple because you haven't yet washed off the crystal violet from the gram-negative. That is only accomplished by adding the alcohol or acetone, that's the decolorizer. So for the first two steps, all the cells are going to be purple. In the third step, once you've applied the decolorizer, the gram-negative will now appear colorless and the gram-positive will be purple. So in order to be able to visualize the gram-negative, then you have to apply the counterstain, which is the safranin, that will allow the gram-negative to appear pink and the gram-positive to appear purple. You may also have a question that says, what happens if you leave the decolorizer on for too long? I think the decolorizer is usually on for like 15 to 30 seconds. It's not that long. And again, you wash with water between each one of these steps. If you leave the decolorizer on for too long, it will remove the crystal violet from the gram-positive as well, and they would appear all colorless. So it's actually kind of a sensitive process to make sure that you get the staining correctly applied so that you can visualize the gram-positive and the gram-negative. So I'm going to actually just write that in here just so you have this correct here. You have a water rinse between each one of these steps. And after. So then once you're done with the gram staining, you would then allow the staining on there to dry, and then you can look at it under the microscope. And the 100x objective requires oil immersion in order to see the specimen clearly. Which would look like what we showed in this picture. So this is actually under a compound microscope or light microscope. This is 1,000 times magnification of the bacteria. So you can see that they still look really small even in that diagram or that picture. It's not a diagram, it's actually a picture of a microscope image. Oh, I forgot I put this in here. So this one is actually from one of our actual labs, our previous lab. And this is or actually it might be a picture. I take that back. But anyways, I like this picture because it shows you the difference in size between eukaryotic cells and bacterial cells. So this pink cell here, put a pink border around it. This pink cell here is a cheek cell, a human cheek cell. And these little bitty purple are gram-positive cocci bacteria. So hopefully that picture really helps you to understand. We looked at the chart that says relatively how big these cells are compared to each other, but this shows you how tiny bacterial cells are. All right, I talked about some of the different special stains. This acid-fast stain is one example of that. And this is used in acid-fast organisms, which I mentioned earlier. They contain mycolic acid on their outer layer that causes them to resist staining. So therefore, they have to have a special process that occurs. In acid-fast staining, bacteria are going to retain the carbolfuchsin stain that is going to appear red against a blue background. And usually with this special staining, so for example, this acid-fast staining, which is also called the Ziehl-Neelsen stain, there has to be heat applied when the carbolfuchsin stain is applied. So this is a picture showing you that. You have the red, just go back again here. Red against the blue background here. So this is showing you that you can actually see the Mycobacterium tuberculosis where these little points are pointing to. And then the blue cells would be the human cells from the sputum. Right. So we left off, we finished up talking about, I'm trying to make this just bigger in PDF to try to use this, so hopefully this will work. We finished off talking about the staining of acid-fast, which you're going to look a little bit more at staining techniques tonight. And the last section gets into culturing bacteria and doing different tests on them. So culture and sensitivity testing is really important, especially because of the fact that some bacteria develop antibiotic resistance, which we were just talking about. So you can have a certain species of bacteria that you would normally treat with penicillin or whatever type of antibiotic, and that particular individual may have acquired one that has developed antibiotic resistance and it no longer is affected by that antibiotic. So it's important to test, especially certain species that tend to mutate more rapidly and pass these strains. So we talked a little bit about collecting samples last time, so it's important. Oh, and I did verify, somebody had asked about a question on, I believe it was the MindTap pre-quiz that was asking about plating organisms in a certain way, and the answer was actually inoculate. So in case anybody's confused about that one, to inoculate is just to move a sample to a different location, basically. And we can inoculate them onto a solid agar plate, or we can inoculate them into a liquid medium. It just depends on what you're going to be doing with the sample. Yeah. So there are certain types of growth medium that have different indicators in them. So for example, if the pH changes, if it becomes more acidic, it may turn from red to yellow, or there's all kinds of different ones. So these are those indirect methods of testing for the presence of certain microbes. Because like I mentioned, we have 6,000 species of bacteria that are in 3 different shapes. So we can't just do the gram stain and say, okay, now we know what it is, because there's still going to be thousands of different species of bacteria that could be. Granted, there's certain ones that affect certain body regions, and so you have an idea of which ones it's going to be, but you still need to do additional tests in order to tell, because they're too small to be just seen under a microscope. And then these are always, and this is one of the reasons that tests sent to a lab aren't analyzed and the results available immediately, is because generally speaking, it takes 24 to 48 hours of incubation after the sample has been inoculated before you can actually read the results. So that's really important. We're going to talk about how you can test for specific antibiotics in a second here. We used to do a lab, I'm trying to remember, it was the control of microbial growth lab, but I don't, it's not in your plan. It's not in your plan this session. They're doing other ones. So, but I'm going to still show you the topic and learn about it, because you still might need to know that material. Okay, so a growth medium is something that contains different things in it depending on what type of bacteria you're selecting for. So for example, we talked about staining last week. We talked about differential staining, where certain organisms are going to stain one color and certain organisms are going to stain another color. You can also have medium that is differential, where it will turn yellow if it's creating an acidic product, or it will stay red if it's not producing an acidic product. Or you can also have selective media, which is actually selecting for or against. Selective. So this will actually either prevent or encourage the growth of certain bacteria. So if they grow at all, then you know that they are the type of bacteria that require that particular ingredient that was put into the selected media. This can be, why is it now? Oops, I started typing into the chat. Or encourage. So certain bacteria can't live without certain amino acids. So we have, this is kind of going off topic, so I'll try to say it quickly. We have 8 essential amino acids that our body cannot make. So we have to ingest them in our diet in order to get those amino acids. That's what we used to call a complete protein, which you don't have to have it all in one meal, but these are amino acids that we cannot make. So certain bacteria have that same issue. There's certain amino acids that they need in order to build the proteins that they need, and they have to get them from their diet. So we can either deprive these of those, or we can add them to in order to prevent or encourage growth. Okay, so in a Petri dish, we're going to use a solid agar, and agar is a substance that's almost kind of like a jello, maybe a little bit more solid, but it's made from an isolate from red algae. So it kind of inherently has a lot of nutrients in it. And then we can adjust what we add to that in order to encourage what we want to accomplish. I believe it is in your week 3 lab, which we'll talk about a little bit more next week, but you're going to have to do bacterial isolation, where you're going to have to separate 2 different species of bacteria from a sample. I think it's from a cerebrospinal fluid sample in a patient in the labster. And you have to be really, really careful and diligent with the streaking method in order for them to separate enough. Because if you think about it, you're actually trying to separate these little tiny organisms from each other that are all kind of clumped together in this liquid, and it's really hard to get them separated. And then beyond that, as individual cells, they're separated, but then they're going to replicate by binary fission over 24 hours, and you have to be able to allow them to replicate long enough for you to be able to see the whole clump of them, which we call a colony forming unit. And so they have to be separated enough that when they form these colonies, they're not overlapping, because if they overlap, you can't take a sample from that one because you don't know if it's just that one bacteria or if it's the 2 different ones. So it's, I'm giving you a heads up for next week. It's a little bit tricky trying to separate them for next week's lab, so you might have to do the streak plate method a couple times. So that's my heads up for that one. And then the other thing that's important in real life about doing these samples or incubating them is that you have to place them upside down in order, which I have a picture of it next here. So I'll just show you this. So we say agar side up. So the lid is not facing upward like you would normally place something with the lid up. You actually place it the opposite direction so that you can kind of see the agar on here. The agar is on top because in the incubator, condensation will build up on the lid, and if it drips down on the sample, your separation is basically shot, because now you've mixed it all together, and you can also get contamination from off the lid. And so it's really important to incubate them upside down. Other important thing for real life application is that you want to write any labeling on the underside where this check is, because the lids on these agar plates are not tight and they spin around. If you label anything on the top, especially if you have like quadrants delineated for what samples you put in what area, that if that lid spins, then your labeling now no longer applies to where you put the sample. So you always want to label things on the bottom. All right, so like I mentioned, usually we do 24 to 48 hours of incubation, and most of the time you're going to be working with a mixed culture because you are taking a sample from a patient in order to try to identify what the infection is, but you have to isolate them into pure cultures because you can't do testing on more than one type and feel like you know what the characteristics are of that one type. So we always have to isolate them, and then you can run the tests on them in order to identify them. So perhaps this lets you know why sending something off to a lab is not something immediate. It's a little bit different when it's like antibody testing, like for viruses, where you just have a little color indicator that says, yes, this is here, or no, it's not. But when you're working with bacteria, you have to culture them for a while in order to be able to get the results. All right, in this PDF, I have a video for the streak plate method. And like I said, you're actually going to do this in lab next week, but this is in this week's curriculum. So we're talking about it now. But this video explains it really, really well as far as how you do a streak plate method. But I'll just kind of explain it a little bit here. Actually, I need this line here. So the way that you do it, it's really important to sterilize whatever you're using to get the sample of the bacteria between each streak that you do. But it's called the streak plate method because you have 4 different quadrants that you streak. So say, for example, we had a test tube here. Here goes my wonderful drawing again. And here's our loop that we're taking in to get the sample. And it's basically just a wire that has a little circle in the end, kind of like the little bubble makers that you used to blow into, except for it's really small. It gets a sample of the liquid. And then you're going to take that and you're going to streak it. It's really hard to do this without overlapping. You do not want to overlap lines like my first line did there. Because as you go further and further, more passes, you're having less and less bacteria left on the loop. And so the idea is you're gradually diluting them so that you have less and less bacteria. So then after that pass, you would take the loop and you would flame the loop in a Bunsen burner. That's supposed to be a flame to sterilize it. And then, so that kills any bacteria that are left on the loop. And then you come across this second, you turn it a quarter turn, you come across the second one, and then you go back and forth, again, trying not to overlap. And then after a couple of times I'm like pointing to the screen, like you can see where I'm pointing. After you cross into the previous streak 2 or 3 times, there's different techniques. And if you know how concentrated your original sample is, you can adjust it. But once you've gone into that previous pass a couple of times, then you continue to do the zigzag back and forth without crossing over that second pass again, because you don't want to get those more concentrated ones. Then you would flame the loop again. Then you would turn another quarter turn, same thing. Most of the labs where I learned, you go back across the previous sample 3 times, and then you continue back and forth without crossing it. It's hard to do this with my mouse, so it's a good thing that the drawing's on there because my drawings are not that great. And then you do that a 4th time. So you can watch that video in order to learn how to do it. It's kind of the same problem with the lab stir because you have to use the mouse to do it. And so you've got to be kind of careful, unless you have a stylus, I guess, that you could use on your computer. Might be a little bit easier to do it, but that makes it a little bit more challenging to get them to actually separate. Okay, so like I mentioned, there are different things that we can use, different types of tests that we can do in order to identify them. Some bacteria have a very distinct look to their colony morphology, which we're going to talk about here in a second. We don't have any pictures of it in here, but it's in the lab that I'm going to show you. Actually, I'm trying to remember. I will figure out for sure which lab PDF it's in, but I'll show you the different colony morphologies. The growth requirements, like I was talking about, different amino acids or different sugars they can use, things like that. Biochemical tests, which would test for them to use some reagent and create an acidic environment. Or we can also do genetic tests where we can actually sequence the DNA. Those would be a little bit more involved. So like I mentioned, these may require several days in order for all these tests to be done. All right, and then the susceptibility testing is getting to the idea that I was talking about, as far as we know that, okay, now we have the species of bacteria. We know that this antibiotic is normally effective against that bacteria, but we may have an antibiotic resistant strain of that bacteria. So we are then going to do a susceptibility test, which is often referred to as the disc diffusion test or the Kirby-Bauer test. Kirby-Bauer disc diffusion test. We also don't do this lab anymore, which is actually probably good because it was a really, really complicated lab. But there was also a serial dilution method, which is a way to take a really concentrated sample and just dilute it out, and then culture each one of those dilutions and do a cell count in order to try to figure out how many bacteria are in the infection. So this helps to identify the concentration of the drug that's needed. So it's, I guess, a relatively simple process, but the actual calculations used to always drive my students crazy. So. All right, so the disc diffusion method is basically where you take this filter paper, it's this little circular disc, and it's soaked in either some type of disinfectant or sterilant or antibiotic, depending on what you're trying to test. And then it is actually first you culture, we call it a lawn of bacteria. So this is different than, I'll put inoculate, that way you see that term again. You will inoculate a lawn of bacteria. So this is different than the streak plate method, because in this technique, you want to, I'll use this little crosshatch, you want to actually cover the whole entire surface with the bacteria. So usually this is done with like a sterile cotton swab or something like that, or you can do it with a spreader, which is actually what you use in Labster. It's this little glass rod that has a horizontal piece at the bottom that you turn in a circle to spread the bacteria all around. But you want the whole entire thing covered, which I'm not doing a very good job of showing that, but I was trying to demonstrate that with the shading. So first you put the lawn of bacteria on there, and then you place this soaked filter paper on there, and then you incubate it for 24 to 48 hours. And if the antimicrobial agent is effective against that bacteria, there will be what we refer to as a zone of inhibition, which basically just means that the bacteria can't grow there. And so you will see a clear section, whereas the rest of it kind of looks hazy where the bacteria are growing, which I'll show you on a picture on our next slide here. So these are the disc diffusion method samples. So you can see here, each one of these samples have a different species of bacteria on them. And all of this kind of hazy white substance is the lawn of bacteria. That's the bacteria growing. And then the zone of inhibition is this clear space around it. And what we normally do is, in order to compare the effectiveness of the different antimicrobials, we would actually take a ruler and measure the diameter of the zone of inhibition across. And then you can say chlorine is more effective than phenylphenol, which is about the same, maybe just a little less effective than quat, and so on and so forth. So you can identify, we can do this for antimicrobial agents like these, or we can also do them with antibiotics in order to develop or determine whether the bacteria is susceptible against antibiotics, which is usually the case you're going to do if you have a patient's sample. You're not going to be treating them with chlorine, you're going to be treating them with an antibiotic. So this disc would instead contain penicillin or erythromycin or whatever on it instead of chlorine. But we can do it for either type of test. There's more specific tests that are similar to this that you can do. These are the minimal inhibitory concentration test, and then also the minimal bacterial concentration test. And so these just kind of get a little more specific than just having the disc coated or soaked in one concentration of the substance. It actually has one end of the stick where it's less concentrated and one end where it's more concentrated. And then you can actually see the change in the growth across. This is a screenshot from the Labster where we did this test, where we, this is the lab that you don't do anymore, but this is showing you a strip here. Get my square back. So this would be a strip that has these different concentrations of penicillin on it. And the minimal inhibitory concentration would be right here at the very bottom, where it just starts to inhibit growth. I had actually created this screenshot because one of my students was like, I cannot get this to allow me to progress. And it was because they weren't clicking low enough. They were clicking up here in the red, and the minimum inhibitory concentration is at the very, very bottom. So but this one over here, the AMT ring on the left, shows you some different antimicrobial agents that might be used. And in this particular Labster, it's showing the zone of inhibition by this red shaded thing instead of a clear circle, but that's representing that that's the zone of inhibition. So in this example here, penicillin would be the most effective against this particular bacteria because the zone of inhibition is the largest. So if you get any questions like that, we used to have them on our other tests, so I'll just mention this just in case. If you get any questions about the zone of inhibition, if the measurement is larger for the zone of inhibition, that means the antibiotic is more effective. And if it has a smaller zone of inhibition or no zone of inhibition, like this chloramphenicol for here has no zone of inhibition, then it is not effective against that bacteria. And that bacteria would be resistant to that. All right, and then in reality, treatment begins before you actually do all of these tests. You got to start with something. We know that these lab tests can take a few days and you don't want the person just getting worse. So treatment begins before these actually occur. And then the type or location of the suspected infection is kind of used to guide that. And then hopefully, if it is a bacteria that has less resistant patterns, then you can kind of proceed with that. But if it's not being effective, then this is when they might turn to the lab results for more assistance in finding something that is more effective. And there are actually certain bacteria, which actually I think it's on the next slide that it talks about, that are called, I'll go to the next one and then come back. Oh, it's 2 away. Here it is. That are called superbugs that are resistant against all of the antibiotics that we have to throw at them. Sometimes in combination, they may be more effective, like a synergistic effect. But if someone has a bacterial infection and it is widespread substantially, and there is no antibiotic to treat them, it actually can be life-threatening. All right, so this the next slide here is just very, very general signs and symptoms for bacterial infections, which of course can be different depending on where the infection is. But if it was a systemic infection, for example, whoops, I keep clicking too far. They could have fever, they could have swelling. Fever is a good indicator that you have an infection because your body is resetting the temperature because it's responding to the presence of a bacteria. Pain, tachycardia, rapid breathing, or abnormal drainage. Yes, so MRSA is methicillin-resistant Staphylococcus aureus. I'm actually not sure. I know there have been many people that have died from a Staphylococcus aureus infection or had amputations or become paralyzed because they had like a infection in their spinal cord, and the removal of the infection caused the removal of the nervous system. So it just depends. There can be different strains of MRSA, but it seems like that one is the most common one that they actually cannot find any antibiotics to treat it with. But MRSA stands for methicillin-resistant Staphylococcus aureus. All right, and then a couple other key terms that you need to know, or I should say suffix. I don't know if suffixes is plural. If something is categorized as static, whether it's bacteriostatic or fungistatic or microbistatic, which would be more general, this means that it slows growth or stops growth. So it doesn't kill it, it just prevents it from replicating more. Whereas anything that is cidal, bactericidal, for example, a virucide is going to destroy the virus, is going to kill it, or in the case of a virus, inactivate it. And then you have different classes of medication that can be broad spectrum or narrow spectrum. Broad spectrum is going to be more effective against a broader group of broader genuses, broader species of bacteria. And narrow spectrum is going to be more specific for certain strains or certain species. In general, to prevent antibiotic resistance, you would want to have testing if you possibly can and use a narrow spectrum antibiotic because it is going to have less of a detrimental effect on your normal microbiota. And then it's also going to be less of a stimulus for antibiotic resistance to develop. So depending on what the infection is, they might start off with a broad spectrum antibiotic, and then if they find that it's not being effective, they may do the tests and switch to a narrow spectrum. Then other specific requirements have to do with whether the organism is aerobic, meaning that it uses oxygen in metabolism, or whether it's anaerobic. And then I talked about the superbacteria, which are those that are resistant. So just kind of some other notes that I put in here about the development of antibiotic resistance, because it actually is a huge problem. And I think it's kind of being ignored, really. I don't know that much is being done about it. You guys can write back to me and let me know whenever you get into core and or when you get out in the field, and let me know whether there is discussion about preventing antibiotic resistance or not. But I found this startling statistic that says 1 in 3 outpatient prescriptions, and this was specifically talking about antibiotics, are unnecessary. There are, and I've seen this statistic many, many times, 1 in 3 patients that are prescribed antibiotics have a viral infection, and the antibiotic is going to do absolutely nothing for them. So it's one of those things. And then this chart here on the bottom is kind of speaking to the spectrum of activity. So the ones that have, so across the top here, you see Mycobacterium, gram-negative, gram-positive, chlamydias and rickettsias, and then it goes into protozoa, fungi, helminths. And so it has across how many different organisms is this antimicrobial effective. So these would be like more broad spectrum. Tetracycline is considered one of the most broad spectrum drugs of choice. And then these that only cover across a few would be a more narrow spectrum. I just closed the chat and somebody typed in it. So